Okay, hello everybody. Welcome to our Biozyme webinar Wednesday. My name is Monika Burbach and I'm a product manager for biochemicals at Biozyme and have been working in the field of molecular biology and biochemistry for the last 20 years. It's so great to see that so many of you could join us for the webinar. I'm very happy that Dr. Arne Lundin from BioThema is with us today. He will give us insights into the quantification of enzymes and metabolites with rapid bioluminescence assays. At the end of the session, there will be time to have a discussion and answer your questions. Please type in your questions into the chat box whenever you like. We will answer them at the end. Thank you in advance for your input. Okay, that's all for me now. Arne, please feel free to make a start.
Hello everybody, my name is Arne Lundin. I have devoted my entire professional life to analytical applications of firefly luciferase, starting in the beginning of the 1970s at the National Research Institute. I worked at Stockholm University and the Karolinska Institute, where I took my doctor's degree. At LKB Products, I had my own product line, and I also started labs at Amersham International and BioOrbit. In 1987, my wife and myself started our own company, BioThema. I have written more than 70 scientific papers and several patents. So that was a short introduction of myself. Thank you very much. Hello everyone. Today's presentation will first describe the basic principles of ATP monitoring using the firefly luciferase reaction. Thereafter, I will give examples of assays that can be performed with this technology. Some of these examples will be described in more detail to illustrate special tricks which can be used to simplify measurements or calculation of results. What are the characteristics of continuous ATP monitoring with firefly luciferase? ATP can be continuously monitored in ATP forming and ATP depleting reactions in a single cuvette or a single microplate well, simply by adding ATP Reagent SL, where SL stands for stable light. Thereafter, you can continuously monitor the ATP concentration by measuring the light emission. The method can be used, for example, for assays of enzymes and metabolites. ATP depletion by the luciferase reaction is low, less than 0.9% per minute, so you are essentially looking at the reaction you want to follow, even though you do it utilizing the luciferase reaction. There is a wide linear range from 10 to the minus 12 to 10 to the minus 6 moles of ATP per liter. If stored in a refrigerator, reconstituted reagent lose only half the activity in 3 months and can still be used if calibrated with an ATP standard. What are the advantages of continuous monitoring compared to assaying aliquots at several points in time? One is obvious, it's less reagent consumption, and you get a more detailed monitoring of the ATP formation or depletion. And you will see that you have reached the endpoint of the reaction. And kinetic deviations are easily observed. And how do we achieve suitable conditions for ATP monitoring? The luciferase reaction consumes ATP and light is formed, and the light intensity is related to the ATP concentration. So conditions for ATP monitoring is ATP depletion in luciferase reaction should be negligible, and inactivation of luciferase during reaction time should also be negligible. And you need a constant temperature throughout the measurement. At ATP concentrations much less than km for luciferase, the light emission V is proportional to S, which is the ATP concentration, according to the Michaelis-Menten equation given here. Stable light simplifies calibration with ATP standard. When you add the reagent to the sample, you get a light corresponding to the ATP level in the sample, and then you add ATP standard and get the light from the ATP in the standard. And there is a simple formula by which you can calculate the ATP concentration in the sample. Just divide the light from the sample with the light from the standard and multiply with the concentration of the standard. There are several advantages with ATP standard calibration. Luciferase is inhibited by several substances, by ionic strength, by deviations from the pH optimum, and by deviations from optimal temperature. Luciferase can also be inactivated, for example by contact with surfaces or by aging. D-luciferin can be degraded by light, oxygen, and racemization. Calibration by measuring the light with and without an added ATP standard compensates for all these problems, as the light emissions from the sample and from the standard are equally affected. Furthermore, as shown in the previous slide, there is a simple formula allowing the calculation of ATP in chemical units, for example picomoles, rather than in RLU, relative light units. This means that ATP measured with a partially inhibited or partially inactivated reagent can be directly compared with other ATP reagents or other techniques. This is not true for RLU. A standard curve with several ATP concentrations is only required when setting up the assay, and then mainly to find the upper limit of the linear range of the luminometer. As the light signal is strictly proportional to the ATP concentration, a standard curve for each experiment is just a waste of reagents and labor. How do you add the ATP standard? When monitoring ATP formation, the standard is added as the last step. The addition may not change the conditions for the luciferase reaction. Consequently, the addition should be made in a small volume, not to dilute the reaction mixture more than marginally. In ATP formation assays, the standard should provide an ATP concentration at least 10 times higher than that of the highest concentration among all the samples. In my illustrations, this has not been possible to do, or the ATP forming reaction would have been difficult to see. The light emission is strictly proportional to ATP at less than 1 micromole per liter, so the same ATP standard concentration can be used in the entire series of samples. So what type of assays can you do with the ATP reagent SL? The ATP formation in kinetic assays is measured and calibrated with addition of ATP standard. And what you measure is the slope. In endpoint assays of metabolites, the ATP formation at the endpoint of the reaction is measured and calibrated by the addition of ATP standard. In kinetic ATP depletion assays, ATP is added first, and then the starting reagent is added. A plot of log of the light versus time will be a straight line with a slope equal to the rate constant of the reaction. In endpoint assays of metabolites, the metabolite concentration must be lower than the ATP concentration, so that only a fraction of the ATP is depleted. There are some considerations when setting up ATP monitoring assays when there is a choice. In general, assay conditions, for example pH, should be optimal for the adenylate reaction rather than for the luciferase reaction, as variations in the luciferase activity are compensated for by the use of the ATP standard. Outside the interval pH 6 to 8, luciferase is slowly inactivated. In case pH optimum of the adenylate reaction is outside this interval, choose the pH optimum of the combined adenylate and luciferase reactions. That is the maximum light signal. Make sure that the addition of the ATP standard does not change the pH or ionic strength of the reaction mixture in a significant way. Here comes some examples of ATP formation assays. We have enzymes like pyruvate kinase, adenylate kinase, creatine kinase, isoenzymes, farnesyl pyrophosphate synthase, and metabolites like ATP, ADP, AMP, phosphocreatine, pyrophosphate, and the creatine kinase can be measured total or as the B subunit activity. Here we see the reaction. The adenylate kinase reaction would form a high blank for the measurement if not inhibited by AMP and the adenosine pentaphosphate. The figure shows the automatic procedure. And the adenylate kinase activity is measured here, CKB here, and CK total here. The CKM subunit inhibiting antibodies allows determination of CKB subunit activity specifically. And this activity is strongly correlated with acute myocardial infarction. The bioluminescence assay was optimized to give the same results as the standardized spectrophotometric assay recommended by the clinical chemistry societies in several countries. For example, pH 6.7 is used. The two methods are strongly correlated. We have also used the CK assay for screening for Duchenne muscular dystrophy, DMD, which is a fatal X-linked disorder with an incidence of 1 in 3,600 boys. As many other inborn metabolic errors, it can be detected in dry blood spots. The US FDA authorized the first screening test for DMD in 2019, as treatments are now available for some forms of DMD. In the test we developed in 1986, the first step is to use the A key from the erythrocytes to degrade the ATP in the dried blood, as shown by this formula. So we can see the degradation of ATP here, and then we add the substrates and measure the increase of the light and then the ATP standard. This test has been used to screen over 500,000 boys, mostly 4 to 6 weeks old, in Germany for high levels of creatine kinase. Follow-up testing by DNA methods will determine type of DMD. We can assay ATP, ADP, and AMP in a single cuvette. When we add luciferase, we get the light corresponding to ATP. Then we add PK to convert ADP to ATP and get a light signal. And then PPDK and get the signal for AMP. The reactions are shown over here. It's also possible to perform the assay using 3 different reagents, one for ATP, one for ATP plus ADP, and one for ATP plus AMP. And from these measurements we can calculate the energy charge, which is ATP plus half ADP divided by the sum of all three. And this is a valuable measure of the energy status of living cells. And here are some examples of ATP depletion assays. Protein kinases, aminoacyl-tRNA synthetase, ATPases, and metabolites like glycerol, urea, and we actually calibrate our ATP standards using a glucose standard and hexokinase. This slide illustrates the Kinetic ATP depletion assay of protein kinases. We used protein kinase A, pKa, and chemtide as substrate. Light versus time was measured from 0 pKa, the blank, up to 107 nanogram pKa. The ATP depletion for the blank is very low, less than 0.06% per minute, corresponding to a half-time of 14 hours. The readings for wells containing pKa were normalized by dividing with the corresponding readings for the blank. On a logarithmic axis, the normalized light versus time was straight lines, providing the rate constant all the way down to the electronic noise of the luminometer. Plotting the rate constant versus pKa level was linear over 3.5 orders of magnitude. In other experiments, IC50 for inhibitors and optimal substrate concentrations were easy to determine. Z' is a parameter used to evaluate the reliability of high-throughput assays. Its highest concentration is 1, but in high-throughput screening it is accepted to be as low as 0.5. When determining our assay, it could be as high as 0.96. In high-throughput screening, the compound libraries may contain millions of compounds. These libraries are screened for substances that inhibit various enzymes. When the inhibition is high enough, the compound is called a hit. In the pharmaceutical industry, high-throughput screening is used to find new drugs towards a wide range of diseases. Consequently, there are several kits on the market, including those based on ATP measurement. The present slide is intended to explain the advantages of the BioThema kinase RR kit. In the paper we published in 2008, the data is based on protein kinase A, described in the previous slide. The method has also been tried for other ATP depleting enzymes, for example aminoacyl-tRNA synthetase. When you plot log of the light emission versus time, you get a straight line as described in the previous slide. So this is how the control will look. If we have pKa inhibition only, then we get a lower slope, while if we have a luciferase inhibitor, we get less light but the same slope as the control. And if both pKa and luciferase are inhibited, we get a lower slope and a lower light. So decreased slope indicates a hit, an inhibitor of pKa. Measurement of light at 2 points in time is required, but no follow-up testing is required. If you measure light at only 1 point in time, then you will still see the pKa inhibitor. But readings below the control, below the green line, cannot be interpreted. And if both pKa and luciferase are inhibited, the rate is uninterpretable or underestimated, that is the dashed line. And follow-up testing will be required. So features for the kinase RR kit. It's feasible for protein kinases, ATPases, and aminoacyl-tRNA synthetase. No interference from luciferase inhibiting compounds or variations in ATP concentration. All library compounds can be classified without follow-up testing. The kinetic assay of glycerol by ATP depletion rate is illustrated in this slide. Lipolysis is the process of triglycerides degraded to free fatty acids and glycerol, and then glycerol is measured in an ATP depletion assay. K is the rate constant of the ATP depletion, and we have tried this at 3 different ATP concentrations, 1000, 100, and 10 nanomoles per liter. And these may look different, but if you plot them on a logarithmic scale, you see that you will get the same slopes, the same rate constants with all ATP concentrations. So ATP concentration is not important for the assay. This assay of glycerol has been used for studying lipolysis at the Karolinska Institute for over 3 decades. As the slope, that is the rate constant, is measured, the assay is neither affected by variations in ATP concentrations nor in luciferase activity. The principles in this slide are used for the glycerol assay at the Karolinska Institute since 1989. Here is the Michaelis-Menten equation, and here are the parameters. And here comes the graph of the Michaelis-Menten equation plotted in as V versus substrate concentration. If you go in at half the maximal velocity, you can come down to the Km value, 40 micromoles per liter. Here, as the insert, we have studied the very, very low portions with very low concentrations of S. The blue points represent where S is lower than 1% of Km. The trend analysis show an acceptable deviation from linearity up to the green circle. The brown points are above 1% of the Km, and you can see that R2 and the slope are lower and that the intercept with the y-axis is higher. These data points are outside the linear range of the plot. The upper limit of the linear plot is 0.4 micromolar. If you instead plot the two sides of the Michaelis-Menten equation against each other, the plot will be a straight line from 0.00 to 1,1. For the glycerol assay, the linearity is up to 100 micromole per liter. This is 250 times better than the conventional V versus S plot. This log-log plot is an alternative depending on which glycerol levels are of most interest. The principles in this slide applies to all enzymatic assays for which the Km value is too low for the substrate levels to be determined. It is a general way to linearize assays based on enzymes that follows the Michaelis-Menten equation. Here are some examples of ATP monitoring of complex reactions. Cell lysis, lysis of bacteria by phages, signaling by leakage of ATP through special holes in cell membranes, platelet aggregation, pyrosequencing, photophosphorylation, oxidative phosphorylation. So oxidative phosphorylation we can monitor using mitochondrial suspensions, isolated mitochondria. And mitochondria supplied with substrates will synthesize ATP, which we can measure. The mitochondrial rate of ATP production varies with substrate supplied and inborn enzymic deficiencies. So the features are that mitochondria isolated from 25 milligram muscle are provided with different substrates for mitochondrial production. Each assay is calibrated by addition of a known ATP standard. The rates with different substrates are used to diagnose which mitochondrial enzyme is deficient. This assay is used in the diagnosis of inborn metabolic errors in small children at the Karolinska Hospital in Stockholm since 1990. Conclusions. ATP monitoring is a highly versatile tool for assays of many enzymes and metabolites, as well as cells and organelles releasing ATP. The way to perform the measurements and calculate the results have been described in this webinar. ATP forming reactions should be calibrated by measuring the light before and after adding a known amount of ATP standard. ATP depleting reactions should be calibrated by measuring the light in the presence of ATP standard. Subsequently, a starting gradient is added and the decay of the light is monitored. No need for ATP standard curve except once for establishing the linearity of your luminometer. We have started to produce application notes. The quantification of enzymes and metabolites is available at the home site. Monitoring of cell lysis, platelet aggregation, and photo and oxidative phosphorylation is coming soon. Quantification of protein kinase and other ATP depletion assays is also coming soon. Thank you for participating in this webinar. Whenever you are about to set up an ATP assay, you are welcome to send me an email. I will be happy to share my experience in luciferase-based assays. The more details you provide on what you want to measure and how your lab is equipped, the more qualified answer you will get. I also look forward to your questions right now.
Yeah, thank you, Arne, for your interesting presentation. So, also, if you are located in Germany and Austria, feel free to contact us at BioThema for more information. Yeah, and now I think we are ready to answer some questions. Just type them into the chat box, and I already see some. We start now with at which temperature can continuous ATP monitoring be performed, Arne?
The temperature optimum is 23 degrees for the reaction. And you can also use lower temperatures, but then you will get less light. And at temperatures above the optimum, there will be some inactivation of luciferase. We run our QC assays at 25 degrees, and a new reagent which would allow running assays at 37 degrees is now being evaluated.
Well, that sounds good. And I have an additional question for you. It's about the volumes. What final reaction volumes can be used?
Well, the number of photons emitted per minute is obviously proportional to the volume of the reaction mixture. Only part of the photons emitted will reach the detector, depending on the geometry of the measuring chamber. At least with some luminometers, the measured light signal is essentially proportional to the volume of the reaction mixture. In the high-throughput screening of protein kinase, the final reaction mixture is 50 microliters, so you can do it in a microplate. In these assays, the luciferase activity and the ATP concentration do not influence the measured rate constant. FDA considered setting up these assays in a reaction volume of 5 microliters, but that requires special dispensing equipment.
Mm.
In our other QC assays, we use 1 milliliter reaction volumes and a temperature control luminometer to achieve the highest possible accuracy.
Okay, so thank you, Anna, and I think now our time is over. So thank you
Thank
to
you.
everybody. And yeah, you also could visit the webinar again on our website in a few days. And I want to say thank you to all the participants and thank you, Anna, for the great talk.
Thank you.
Bye.
Bye-bye. Bye.